A very warm welcome to all of you joining us today. Whether you're dialing in from Europe, the US, or further afield, we really appreciate you taking the time to join us, and it's great to have you here. If you work in drug discovery or development, there's a good chance DILI, or drug-induced liver injury, is a term that has featured in conversations you'd rather forget. It remains one of the leading causes of drug attrition and post-market withdrawal, and despite decades of research, it continues to catch teams off guard. The hardest part is if this happens very late in development, when the cost of failure is at its highest. The challenge isn't just scientific, it's strategic. When do you look? What models do you trust? And how do you make confident go, no-go decisions when the biology is complex and the predictive tools are still evolving? And that's exactly what we're here today to explore. Whether you're designing an in vitro tox strategy from scratch, evaluating outsourcing options, or simply trying to build a stronger scientific rationale for your safety data package, today's session is structured to give you practical, actionable insight. We'll be covering the key biological mechanisms that underpin DILI, and importantly, why understanding those mechanisms, not just observing toxicity signals, are so critical for meaningful risk assessment. We'll look honestly at the limitations of traditional preclinical models and why certain liabilities remain stubbornly difficult to predict. And we'll then explore the in vitro strategies that are helping teams detect risk earlier. And finally, we'll look ahead at where the science is going and what emerging approaches could mean for the field. An expert who understands this space and how to get the most out of it is Sanna Marie. Sanna Marie Atsinki is head of drug interactions and in vitro toxicology at ADME, a Symeres company. She has extensive research experience related to hepatic energy metabolism, gene regulation, mitochondrial function, and toxicity, as well as a strong scientific background in various cell-based assays and mRNA analysis techniques. So without further delay, I'm delighted to hand over to Sanna Marie.
Thank you, Katie, for the kind introduction. I also want to thank the audience for joining today. My presentation is titled DILI Uncovered, How to Mitigate Hepatotoxicity Failure. And I hope it will give you an overview on drug-induced liver injury, its mechanisms, and views on how to mitigate the risk during drug development. This is the content of my presentation today. First, I will give a brief introduction to liver physiology as well as functions. Then we will go through the main mechanisms leading to DILI and some risk factors predisposing to it. Next, we will look into the current preclinical safety approaches, after which I will concentrate more on the emerging novel methods for DILI assessment. And the presentation will end with future perspectives and key takeaways. So first, some basics on liver and DILI. So when talking about the liver, it is one of the most essential and versatile organs in our body. The liver functions include carbohydrate, fat, protein, and hormone metabolism. Liver stores many important vitamins as well as glycogen and iron. Additionally, and relevant to this presentation, one of the main functions of liver is the metabolism of foreign substances, including drugs, and making them excretable from the body. Liver also metabolizes and excretes bilirubin into the bile. Liver consists of the main parenchymal cells called hepatocytes, which comprise about 80% of the total cell amount. Additionally, endothelial cells, Kupffer cells, and stellate cells are the main non-parenchymal cells of the liver. The Kupffer cells are specialized macrophages, very important players in the hepatic immune response. And here you can see a microstructure of the liver with the hepatocytes shown here as these bigger cells surrounded by endothelial cells and other NPC cells. Also, the biliary canaliculi are marked here within the hepatocytes, and the bile duct, which collects the bile from the canaliculi, is shown here on the right-hand side of this picture, along with the branch of portal vein, the hepatic artery, and then the central vein here on the left-hand side of this figure. Then going to DILI. What is DILI? Drug-induced liver injury is one of the leading causes of acute liver failure. It's been estimated to cause about 50% or even more of these cases. The second most common reason is viral hepatitis in about 12% of the cases. DILI develops with variable onset, from mild liver enzyme elevations even up to complete liver dysfunction and fatality. DILI is often challenging to diagnose. Patients may have several co-medications which hamper the assessment. The diagnosis is usually based on excluding other causes. The patient's symptoms and history are key information needed for this. The biomarkers currently used in the assessment include these aspartate aminotransferase, alanine aminotransferase, alkaline phosphatase, and total bilirubin. However, these biomarkers are not fully specific for DILI, nor they classify the severity. Liver biopsy may help in unclear cases, but novel biomarkers are needed for the diagnosis of DILI. And actually, promising candidates have already been identified, such as certain miRNAs and cytokines. What makes liver then susceptible to DILI? It is a highly perfused organ with the portal vein blood flow bringing nutrient-rich blood from the gastrointestinal tract before entering the systemic circulation. The portal flow comprises of about 75% of the total inflow to the liver. Additionally, drug metabolism occurring in the liver may activate the parent drug into toxic metabolites. And due to these reasons, high amounts of the drug and the potential toxicant can be present in the liver and consequently cause toxicity. And here again, you can see the structure of the liver, especially the vasculature structure. So again, the portal vein along with the bile duct and the hepatic artery are shown on the sides of this structure here, and then in the center is the main hepatic vein located. Then going further into DILI. So DILI can be classified into different categories. So first, in this first column, we have the intrinsic or direct DILI. The incidence of direct DILI is high among these categories. It's usually dose-dependent, predictable, and reproducible in animal models. It occurs rather fast, and the cause is really the intrinsic hepatotoxicity when the drug is given at sufficiently high doses. But then, a type called idiosyncratic or iDILI happens rarely. It's usually not dose-dependent at pharmacological doses. It's usually not well predictable or reproducible in animal models. Idiosyncratic DILI develops with varying latency. It can take a very long time to develop, even years, actually. And this is because the mechanism of DILI is often related to an immunological response occurring, for example, after repeated administration. And then we have a third class called indirect DILI, and this is a recent classification of DILI. And the characteristics here are somewhere between the intrinsic and the idiosyncratic DILI. For example, usually it's not reproducible in animal models and does not show classical dose response. The incidence of indirect DILI is intermediate, but it's partially predictable. And this is because indirect DILI mechanism is usually related to the pharmacology of the drug, meaning that it may be exaggerated in terms of the effect and activate the immune system indirectly and, for example, predispose the patient to an underlying liver disease. And known hepatotoxic drugs for all these categories are listed here on the lowest row. So these DILI classifications are important to keep in mind because all potential DILI signs cannot be assessed with the current testing approaches used. And this is a topic that we will come back later in this presentation, but it's good to keep in mind already now. The preceding mechanisms behind DILI have been quite extensively studied in the literature. Here is a list of most common cellular signals. So mitochondrial dysfunction and oxidative stress are key mechanisms occurring in most of the DILI cases. Bile efflux inhibition can result in form of DILI called cholestasis. Also, lysosomal disturbances and phospholipidosis, as well as general excess lipid accumulation or steatosis, are conditions which can lead to DILI. Furthermore, endoplasmic reticulum stress has been linked to drug-induced cholestasis and liver lesions. And as mentioned in the previous slide, immune-mediated responses are key mechanisms, especially in idiosyncratic DILI. Finally, the potential formation of reactive metabolites can result in, for example, adduct formations and consequently hepatotoxicity. However, these mechanisms cannot be considered in isolation from each other. And actually, many of them affect one another, and several of these mechanisms can be involved in a single hepatotoxicity response. Then, let's take a look at some properties related to the drug itself, which are risk factors for DILI. It's been known for a long time already that essentially a dose defines if a substance is a poison. So also here, drugs that are given or planned to be given at high doses or result in high Cmax values are in risk to cause also DILI. Then high lipophilicity or log P is usually predicting higher DILI risk. Fraction of sp3 hybridized carbons or FSP3 within the structure affects the three-dimensional properties of the drug. This is essentially a chemical bonding property within the carbon atoms in the drug molecule. But high FSP3 means greater saturation, improved solubility, and drug-likeness, whereas a low FSP results in lower saturation, more planar structure, and higher risk for DILI. The potential to bioactivate and result in reactive metabolites is a DILI risk property, as also mentioned in the previous slide. Some other physicochemical properties can also be considered as risk factors and predictors for DILI. Here, just a few listed, such as ionization state, polar surface area, hepatic partitioning, the molecular weight, and so on. And with this figure, I wanted to summarize the first part of this presentation, basically on the complexity behind the DILI clinical phenotype and outcome. So if you think about the drug which is given to the, or is going to the body, what affects the outcome is, first of all, the drug properties, the physicochemical, pharmacological, toxicological, and off-target activities. These affect, for example, what is the actual response in the cells, so the mechanism of the injury that can happen. But then at the same time, also the host factors, so the factors in the people, so for example, genetics or age or gender, lifestyle, disease conditions, other medications affect the response. So especially considering what kind of immune reaction is expected. And this can be then, the injury can be also repaired, but it can basically cause the injury, which eventually causes the clinical phenotype. But all these factors are good to keep in mind when you are thinking about the mechanism of DILI happening. All right, but next, let's go through the current and then the emerging approaches for safety evaluation in drug discovery and DILI assessment. Here you can see the drug discovery and development pipeline figure, which is probably very familiar to many of you. So currently, the early in silico predictions are done in the discovery phase when only minimal information is available. Then when going further, also, of course, in vitro studies are done. It depends on the project and the sponsor, what kind of non-GLP assays are done to screen, for example, DILI liabilities. But basically, there are currently no regulatory or GLP in vitro studies that would aim for addressing DILI. The GLP in vitro studies mentioned here usually refer to the genotoxicity or the skin toxicity studies. But consequently, the preclinical DILI assessment relies heavily on the general in vivo GLP toxicology studies. And these studies include studies like the subchronic and chronic toxicity in rodents and non-rodents. And with these studies, the target organ toxicities are identified, for example, liver. And these studies typically take from months to years to complete. And as I explained earlier, when talking about the different DILI classes, iDILI and indirect DILI are not well reproducible in the animal models, but still these in vivo tests continue as the golden standard in toxicology. And furthermore, there are examples from the clinic on issues regarding liver toxicity and failures in the clinic. So this first figure here, they have looked at several organ systems involved in safety failures, and liver is shown here to be one of the most prominent organs for failure at both the preclinical and clinical phase. Furthermore, in this other publication from a few years back, looking at safety issues in drugs withdrawn between years 1990 and 2010, hepatotoxicity was showing the highest incidence with 27% of the cases. In 2009, FDA has published a guidance for the industry for severe DILI evaluation. However, this document concentrates only on how severe DILI can be detected at the clinical phase to avoid the most severe outcomes. The detection is based on the biomarkers ALT and AST combined with total bilirubin. And this combination has been shown to be common in these severe DILI cases. So I've now explained the current approaches of DILI assessment and implied that there are limitations in these. And next, I would like to present solutions to this. And these are the emerging methods and the so-called new approach methodologies, or simply the NAMs. NAMs are 2D and complex in vitro systems, organ on chips, in chemical and in silico modeling methods. The definition of these are that these methods allow moving away from relying on only animal testing in the preclinical safety assessment. The US FDA published last year a roadmap to reducing animal testing, which encouraged the use of NAMs in drug discovery and development, with the initial focus on special modalities such as monoclonal antibodies. Furthermore, this year, FDA has published a draft guidance open for commenting on the general considerations for the use of NAMs in drug development, with the focus on the validation of the NAMs for specific context of use. So a bit more on these NAMs. Why do we need them, and when are they particularly useful? So first, they may identify drug toxicities or mechanisms for which there currently are no adequate models available. For example, special modalities such as monoclonal antibodies are the initial focus, for example, in the FDA roadmap guidance. Then a key benefit is that the NAMs provide more human relevant and predictive information. And furthermore, NAMs can be used as part of the so-called weight of evidence approach, where all information and data available for the drug is considered, and the NAMs can actually fill some knowledge gaps remaining, for example, after doing some in vivo studies. Finally, NAMs can refine the needed in vivo studies, for example, by reducing the durations of the in vivo studies, eventually for all drug modalities. And all in all, the NAMs allow faster and more accurate human risk assessment. And next, let's take a look at different NAM options a bit more detailed. First, the in vitro methods for DILI assessment. So on the first column here, we have the 2D and the monolayer culture models. There can be several different cell types used in this screening phase. Hepatic cell lines can be used, but then, for example, primary human hepatocytes are already a more relevant cell model, having the liver-like metabolic capacity compared to the other cell types. And these cultures, even though they are simple and easy to obtain and culture, they are good for this high throughput screening phase, and they really provide the first hints of potential hepatotoxicity liabilities. And possibly these liabilities can still be designed out in the medicinal chemistry phase. And the endpoints that can be measured or are usually measured from these include, for example, high throughput imaging, giving also mechanistic information, for example, on the mitochondrial impairment or oxidative stress, but also viability, cell death, morphology, or impedance are the usual endpoints measured. But then, as the drug discovery pipeline goes further, the amount of drug molecules or pre-candidates decrease. And then these so-called complex in vitro methods become more feasible and relevant. All these three models described in these three columns belong to these so-called microphysiological systems, or MPS systems, abbreviated here. So, for example, spheroids or microtissues are usually composed of primary human hepatocytes or with co-cultures with the NPC cells, and which assemble into these spheroid-like structures in the plate wells. And they already mimic some liver function and metabolism. And the benefit of these cultures is that these can be maintained for a longer period, and it allows repeated and long-term exposures. And then the organoids, these are self-assembled stem cell-derived liver-like structures. They have multicellular interactions. They already mimic the liver physiological architecture, having the key hepatic functions in them. And finally, the liver on chips. These are the ultimate liver-like in vitro models with high concordance to the in vivo liver architecture and functions. And these systems even have this flow of nutrients coming into the system and out of the system, resembling the in vivo situation also in that sense. However, these liver on chips are not suitable for initial screening. They are quite low throughput and they have a lengthy preparation time. And in addition, they are expensive to obtain and use. The endpoints that are measured in these complex systems include usually viability, which is the measure for the actual hepatotoxicity, but also other functions such as albumin or urea secretion can be measured, as well as the enzyme biomarkers, and then some imaging endpoints as well. Then going further, so to enable these complex in vitro methods to qualify as regulatory tools, the US FDA initiated the innovative science and technology approaches for new drugs, the iStand program, a few years back. And the purpose of this program is to qualify innovative tools and technologies for the use of regulatory submissions to assess, for example, safety, pharmacology, and efficacy. And in the program, the model developers submit the application and qualification plans to FDA for evaluation. The ultimate aim is to qualify the microphysiological system for a specific context of use, which can be, for example, the structural analogs of drugs which toxicity profile is already known. So in these, the reproducibility, robustness, and reliability are evaluated, and this is expected to bring more confidence in the data produced for DILI assessment and for the regulators to accept this data. So recently, the iStand has accepted a liver-on-a-chip technology to be qualified for DILI assessment. But to my knowledge, no DILI MPS model has yet been fully qualified in the program. But this example just shows that there are already proceedings towards the regulatory acceptance of these type of models, also for DILI assessment. Next, let's take a look at some typical computational approaches as part of this NAM concept. So on the left-hand side are the traditional structure-activity relationship models, with QSAR being probably the most used model here. QSAR typically analyzes the molecular descriptors of the drug. So the principle of QSAR is based on the assumption that molecules within similar structures exhibit similar toxicological activities, and then that the physicochemical and structural properties of the molecule can be encoded as molecular descriptors to predict the toxicity of structurally related compounds. Then we have the PBPK or PBTK simulations, which are used for predicting ADME and toxicokinetics of the drug, with possibility of using different physiological models or groups. But in this slide, I wanted to emphasize these two other models. So this is currently the hype in the field of computational approaches are within these various artificial intelligence models. And artificial intelligence is a broad term into which these so-called machine learning models and deep learning models fall into. What is common in these AI models is that essentially they can be taught using a training data set to make then varying toxicity prediction outcomes. So based on the use, the machine learning models or the deep learning models used, so which are listed here, these are kind of sub-models of these wider terms. The algorithm is a bit different in all of these, and the principle of the prediction is a bit different. In general, the deep learning models are used for very complex data compared to the more traditional machine learning models, which utilize relatively simple algorithms in their predictions. But as a summary of all of these computational models, the QSAR, the ML, and DL models are used in DILI prediction already, but mostly at the drug discovery phase to support in-house decision making. There are a high number of publications available and open access algorithms too. And also a few commercial models and service providers are available for in silico prediction of DILI. Then let's discuss a few slides on the potential future approaches of DILI assessment. The future of preclinical safety assessment could look something like this. This is an industry perspective on the FDA Modernization Act 2.0, which aims to reduce the use of animals in drug testing. In this picture, I want to focus on this preclinical phase, which suggests that the in vitro and MPS models could be used for de-risking key target organs, including the liver. Also, AI and ML approaches would be the computational models used in the screening of drug candidates and identifying target organs. Then in vivo studies could be used to complement all these studies. And in the later phase, the lengthy in vivo studies could be refined by, for example, using virtual control groups or reducing the animals and doses in the studies, or by avoiding repeated studies. And also by using the weight of evidence approach, the data gaps at later stages could be complemented using the NAM approaches. Then a summary for the considerations of future DILI assessment. This slide partly summarizes also the previous figure I presented. So in future DILI assessment, the NAMs would be routinely utilized in early drug discovery already, bringing human relevance and efficiency in this early phase and reducing drawbacks at later stages. The drug properties, so for example, the physicochemical properties, would also be utilized routinely in screening and predicting toxicity and DILI risk. The in vitro methods should be used as early as possible, starting from high throughput screening methods and continuing to more complex in vitro models. And then in the regulatory IND and NDA phase, animal experimenting is continuing as needed, but with refinements and in combination with the NAM approaches. And MPS and in silico models are being qualified for use in the regulatory submission with defined and well-justified scope. So as a summary, the weight of evidence approach is the future of DILI assessment rather than relying on single test results. The drug properties, as well as all the data generated, so the in vitro, in silico, and in vivo data, are taken into account in the assessment. It's very important that the performance of the model is known, so the accuracy, the sensitivity, and specificity. And this is especially in regarding the regulatory context. So finally, I would like to summarize my presentation. So currently, DILI assessment relies heavily on animal studies. The extrapolation to humans is insufficient in some cases, and of course, there are ethical issues in using animal studies. The emerging in vitro and in silico methods, the NAMs, serve as promising alternatives, providing potential toxicity information in early DILI assessment or retrospectively. The NAMs are not aiming to replace all in vivo testing yet, but they may be better options for certain modalities, and they may be used in this weight of evidence approach, combining all data available and the drug properties. And while no single assay can reveal all DILI liabilities, it's essential to understand the confidence and limitations of each test, so the accuracy, sensitivity, and specificity, and also the mechanistic information that is possible to gain. So with this, I would like to thank you for this presentation and hand over to Katie.
Thanks, Anna-Marie. As we move into the Q&A part of our webinar, just a quick reminder that you can enter your questions via the Q&A area in the top panel, and the more questions, the merrier. So our first question, what in vitro models would you recommend in, excuse me, what in vitro models would you recommend in preclinical DILI assessment?
Yeah, so there was this one slide about this, these models. So I think there should be a tier-based approach. So you start first in the very early phase, you start screening for the DILI liabilities already with the simple models, just basic 2D hepatocyte cultures, where you can basically flag your compounds already then. But then the further you go in the pipeline, you can then utilize these more complex in vitro methods already in the screening phase, but then hopefully also in the regulatory phase in the future.
Thank you. What is the role of transporters in DILI, and how should we address those?
Yeah, so this is related to the mechanisms of DILI, and I did not go that detailed or that deep into those, but I had the mechanism of bile efflux inhibition. So in this mechanism, the transporters which transport the bile from the hepatocytes into the bile duct are the key mediators. So if a drug inhibits these transporters, so the well-known transporter is the BSEP, so the bile salt export pump, and then some MRP transporters. That can also be a risk for DILI and the cholestasis, basically. So this is the inhibition of bile efflux from the hepatocyte. So also the transporters can be screened for inhibition for DILI liabilities.
Thank you. Does reactive metabolite formation always mean hepatotoxicity risk?
Well, not always. So it depends. For example, I had the figure that the host response also affects the outcome of the toxicity. So there can be some repair mechanisms that can kind of circumvent this injury. So it doesn't necessarily mean it. And of course, if the reactive metabolite doesn't form any adducts that, for example, cause injury in the cells. So that's also possible that it doesn't really cause DILI. However, I just want to emphasize that reactive metabolite formation is a flag for hepatotoxicity and DILI. So I wouldn't take it lightly if I would see that kind of results, but just to be aware of the possibilities.
Thank you very much. Another question that's just come in is, using a tiered approach, starting with 2D models, can you suggest relevance from other models such as HEPA-G and other cell lines? Are these models equally as good?
So comparing the cell lines, well, we have been using many cell types in-house, and of course there are differences in those cell lines. So if you start from, for example, HepG2 cells, and then compare those to, for example, HEPA-RG, I would say that HEPA-RG they have more metabolic capacity. So you could see this kind of metabolite formation, or these kind of indirect effects of if there is a metabolite involved. So in that sense, I would think that the more you have physiological metabolism, the better the model is. So I would say that cell lines are maybe not that preferred, but of course those are good for the initial screening, and then you can go further to HEPA-RG or then primary hepatocytes.
Sorry, I think there's possibly one last question, unless there's any others that appear in the Q&A panel as we are answering this one. But how can mitochondrial toxicity and oxidative stress contribute to preclinical DILI signals?
Yeah, so again about the mechanisms. So I think I mentioned that mitochondrial toxicity and oxidative stress both are one of the key mechanisms behind most of the DILI cases or the hepatotoxicity cases. So these have high incidence among the DILI cases, and for that reason, I think that they are very important to be assessed, and already in the very early phase. And there are many type of assays that can be used for checking those. So for example, imaging-based assays, or the glucose galactose assay for mitochondrial toxicity, and so on. So yeah, it's quite important, or those are quite important mechanisms.
Thank you. Okay, unless there's any other questions that appear in the Q&A panel, I'll give people a couple of minutes. But I'd just like to take the time to thank you, Symeres, for your excellent presentation and the insights that you've shared as part of the Q&A, and also thank all of our attendees for taking the time to attend. And finally, if you enjoyed this webinar, we have a series of webinars available on demand. So you can scan the QR codes on screen and go straight to that page and start learning. So our first webinar was recorded earlier this year with Goran Vespiri, one of our subject matter experts internally, and James Lanter from 8D Medchem LLC on embracing scientific complexity to mitigate toxicity issues and development risks while advancing towards the clinic. And then our second webinar, which was completed a couple of weeks ago, is entitled The Greatest Hits, and it's a beginner's guide to high quality starting points for drug discovery. And that was held by Sam Custers, who's our head of chemistry for Symeres. Thank you all very much for taking the time to attend today, and we hope to see you again at one of our webinars soon. Thank you.